It's my greatest honor to introduce to you our shiny new Professor Yeran Bai. She is currently assistant professor in the optical sciences, and Dr. Bai earned her BSc in optical science and engineering from Huazhong University of Science and Technology in 2013. And then completed her PhD in optical engineering at the University of Chinese Academy of Science in 2019. Then she joined a group in Boston University and became the postdoc there. And she studied the photothermal infrared imaging and its biomedical applications. And then she became another postdoc scholar at the University of California, Santa Barbara, where she investigated metabolic alterations associated with Alzheimer's disease. So let's welcome our speaker.
Thank you for the very nice introduction. It's a great pleasure to be here today and share our research. As Daniel has introduced, we started a lab in the college about 2 months ago, and our lab focuses on developing advanced biomedical imaging modalities and apply them to study important biological phenomena. So throughout my career, I've been always driven by one key question, is that how we can use light to visualize the invisible biological process. And that leads to today's topic, how we can use the mid-infrared photothermal imaging to achieve high-resolution imaging for biomedical research. So let's start it by talking why we need biomedical imaging. So to me, the ultimate goal is to understand the complexity of life, to understand how biological systems function and how they change over time at healthy and disease state, and use that information to improve disease diagnosis, monitor treatment, or even enable new scientific discoveries. So biomedical imaging operates at different scales. And here are a few examples of the different biomedical imaging modalities. The MRI can offer whole organ level imaging in a non-invasive manner, and histology can help us to visualize the tissue level morphology. Taken a step further, fluorescence microscopy can help us see inside the cells or even for single molecule imaging. Today, we are going to focus on one important and very powerful tool in the biomedical imaging field, that is optical imaging. So as I have mentioned before, the biological systems are highly complex. Here's an example of a different neuronal cells visualized by fluorescence imaging in a mouse brain. As you can see, it's highly complex and highly organized. On top of complexity, the biological system is also highly dynamic. Here's an example of a mouse pre-adipocytes filming at 1 frame per second using a phase microscope. Therefore, to visualize the highly complex and highly dynamic biological systems, it requires a biomedical imaging modality to have these properties. Fluorescence imaging, as one of the optical imaging methods, satisfying most of the above-mentioned requirements. It enables highly specific imaging. For example, the sub-cellular organelle imaging inside cells. It also enables functional imaging. For example, using this fluorescence protein, we can visualize the neuronal action potential in this living animal. However, fluorescence imaging requires labeling, and the labels are typically pretty bulky. For example, if our target protein is the human alpha tubulin, as shown here, to visualize that, we will genetically fuse it with the fluorescence protein, here the green fluorescence protein. As you can see, the label here is almost the size, half the size of your target protein. So the bigger size will potentially cause the perturbation to normal physiology. For example, when it interacts with another protein, dragging the whole big label here will cause some perturbation. And more importantly, it's often hard to label some small molecules, which explore us to use this powerful technology to study some important biological phenomena, such as metabolism. On the other hand, the cellular contrast can be imaged without fluorescence labeling. For example, the cellular morphology can be imaged with all these well-established optical imaging methods, such as phase contrast and dark field imaging. Over the years, researchers have also developed other imaging modalities where the contrast is based on more than morphology. such as the autofluorescence and second harmonic generation. So through this technology, we can get more specificity. And among these technologies, the vibrational spectroscopy imaging has emerged as one powerful tool for biology study because it provides very rich chemical information. Now let's take a look at what is vibrational spectroscopy. So as the name suggests, it detects vibration signal inside biomolecules. And we will use the cell membrane as an example. If we zoom into a cell membrane, it composed of a phospholipid bilayer. And if we zoom into one of the phospholipid molecule, it composed of a head group and also two tails. Those tails are fatty acids chains. And the fatty acids have a lot of CH2 bond inside of it. And the chemical bond or the covalent bond connecting the carbon and hydrogen is not rigid stick, but rather they are constantly vibrating. And they have different vibrational modes as shown here. So you can see it can do symmetric stretching, anti-symmetric stretching, or even like a rocking motion. And this vibrational frequency, if you convert it to wavelengths, is lies in a window what we call mid-infrared light. The wavelength is typically on the order of 2.5 to 25 micron. Mid-infrared spectroscopy imaging is one type of vibrational spectroscopy. It's based on absorption. So you have a sample here, you shine a beam of mid-infrared light to your sample, then you measure the power loss after your sample. By changing the input wavelengths, you can get an absorption spectrum out of it. If you're using the broadband IR source, you can get the spectrum from Fourier transform. If you have a narrowband source, you can sweep the input wavelengths to get a spectral out. So the mid-IR spectroscopy find its use in biology because all of the major biomolecules, these are 4 major classes of biomolecules, have their characteristics on the mid-IR spectrum. Taken the protein as an example, it has 2 characteristics. One is called Mi1, which is centered around 1650, and the other is Mi2, which is centered around 1550. Which means that if you shine a beam of infrared light at these frequencies, you can map the protein content in a cell in a label-free manner with high specificity. So the mid-IR imaging has enabled many important biomedical discoveries. For example, if you couple an infrared probe with mid-IR imaging, you can visualize the metabolic process in cells. And if you perform hyperspectral imaging as shown here, and then do the classification based on your hyperspectral images, you can do the virtual histology so that you can use the image for disease diagnosis. Despite it's very powerful, the mid-IR has inherent limitations. For example, because of the long IR wavelengths, the spatial resolution is typically limited to a few microns. This few microns is about the size of a single cell, which means that we cannot see inside the cells. And also, again, because of the wide mid-IR range, the spatial resolution that is dependent on the wavelengths also changes when you tune the IR wavelengths. And because it's an absorption-based method, we will use an IR detector to detect the power attenuation of mid-infrared light, but the IR detectors are typically much noisier compared to visible light. So with these limitations, we ask ourselves, is it possible that we convert the mid-IR absorption into a visible signal that we can detect? Yeah, that's exactly what our innovation is doing. So our innovation is called Mid-Infrared Photothermal, MIP, or Optical Photothermal Infrared, OPTIR. It's a pump probe technique where the IR is a pump and the visible is a probe beam. So let's take a look of how does this technology help us to break the IR diffraction limit. For example, we have a sample. For example, we have a cell sample, and these blue bars are the target features that we are interested in. If we shot in conventional IR imaging, we shot a beam of infrared light to our sample. Because of the relatively coarse diffraction limit associated with IR, we cannot resolve these details inside our cells. On the contrary, if you can focus the visible light much tighter so that we can visualize these features very clearly. But on the only using visible light, we don't have the infrared spectroscopy information. And our idea is that we bring these 2 beams together. And so the process is like this. When we shine a beam of infrared light on our sample,
and our feature absorbs this infrared light, and the absorbed energy will be released in the form of heat. And it will cause a localized temperature increase, and this temperature increase will lead to an effect called photothermal effect, and will cause this sample to expand and also change the refractive index in these localized regions. Then we introduce the second visible light shine to the smaller feature. We know that and the visible light will be scattered off from this feature. And we know that the scattered light is related to the sample dimension and also refractive index. So by measuring the scattering intensity in the far field before and after infrared absorption, we can map the infrared absorption based on our visible light. So by doing this, we can achieve much tighter focus, but this far field scattering can also carry the infrared absorption information. The MIP can be performed in two modalities. One is in spectroscopy mode, where we fix our beam at one location and scan across along the wavelength domain and get a spectrum. Or if we know the spectral feature that we're interested in, we can fix our IR wavelengths and scan across the field of view to create an image. So by doing this, we can much improve the infrared imaging resolution down to sub-micron spatial resolution. And also we can, while getting the much improved spatial resolution, we can also maintain the IR spectral information. And it's also highly compatible with other imaging modalities such as fluorescence. So with this concept, we have built our setup, and this is how it looks like. We have two beams. One is mid-IR beam. The other is visible beam. And the mid-IR beam is modulated at around a few hundreds of kilohertz. And then we combine these two beams together and collinearly shine to our sample using a reflective objective. And the sample transmitted light will be collected by an objective and then sent to our photodiode and to a locking amplifier for sample demodulation. So as a spectroscopy measurement, we first want to demonstrate that MIP can achieve high spectral fidelity. Using two samples, one is polystyrene, another is olive oil. We test the spectral performance of our MIP, and we also compare it with the FTIR absorption spectrum, which is a gold standard in the IR absorption field. So you can see these two overlap pretty good, which means that we indeed can maintain the IR information. Next, we demonstrate that we can demonstrate our spatial resolution by image a 500 nanometer polymer microsphere, and by doing the MIP imaging and then do the X and Y cross-section profile, we quantify our spatial resolution to be around 600 nanometer, which is at least an order improved compared to the conventional mid-IR imaging. So we then apply this system to study the biological samples. The first sample we tested is the cancer cells, and the band we are targeted is the lipid droplets, which is the energy storage organelle inside our cells. So you can see these are 2D plane images, and we also change our objective focus to get a 3D image, and this is a 3D reconstruction of the lipid droplets inside the single cells. We also apply the system to study bigger scale samples. Here's a C. elegans, which is a millimeter long worm. And we are also targeting at either lipid droplets or protein contents inside this living animals. By now, the MIP images are all created by raster scanning point by point of samples. So taking the cell image as an example, it will take about 4 minutes to create a 2D image. And for a bigger sample, it will take much longer time to get a result like this. So this low imaging speed will restrain us for high throughput applications and also restrain us to study the dynamic samples. So we ask ourselves, is it possible to boost the imaging speed? And this leads to our next innovation, that is wide field IR photothermal imaging. And in this geometry, there's no scanning mechanism, but rather the infrared and visible light are all shined to the sample in the wide field manner. Here's our setup. If we don't see the bottom part, that is a mid-infrared part, we only see the top part is really is basically the same as the wide field fluorescence imaging, where we have our laser source and our LED emitter is conjugated to the objective backfocal through a 4F system. And the sample is illuminated with a near parallel beam. Sample reflected light will be collected by the objective and then imaged on our CMOS with the tubeless.
What kind of temperature variations are you seeing?
A few Kelvin increase.
Few
Few Kelvin increase.
few degrees Kelvin.
Yes. Yeah. So we will cover that later in the slide, but that's a very good question. Yeah. So for the bottom part is the infrared light beam pass. We have our air light is modulated with a rotating chopper, and then you can see after chopper we have a pulse train, air pulse train like this. Sometimes we have air pulse on, we will call it hot frame, and the other region we don't have air pulse, we will call it cold frame. When the air pulse is on, for hot frames will cause the sample reflected light to increase. So by detecting the difference between the hot and cold frame, we can create a chemical specific imaging on our sample. Because in the wide field geometry, we don't have a multi-element locking amplifier for us to extract the signal. So we carefully design a technique called virtual locking technique, which basically is we design the electronic synchronization between different instruments. We'll use a function generator as a master trigger to trigger our probe source, our infrared source, our detectors. Here's the oscilloscope acquired image from our pulse trains. You can see we have air pulse on, hot frame, air pulse off, cold frame, and the visible pulse is always on. If we zoom into one of the pulse pairs, the pulse width for the air pulse is around tenths of nanoseconds, and our visible light pulse is around 1 microsecond. So we test our sample. Here is the thin polymer film spin coated on a silicon wafer, and we can see from either hot or cold frame, we don't see too much contrast because it's a uniform distributed thin film sample. But if we subtract between these two frames, we can see a clearly contrast in our center of field of view, which is corresponding to our weakly focused IR spot. Because the IR photothermal is a dynamic process, we have a heating process, we also have a cooling process. So we can map that process by varying the relativity between these two pulses. And we did that, and you can see that we can map the thermal heating and cooling process. Here again, the sample is a thin polymer film deposit on a silicon wafer. And we extracted the maximum intensity from each frame and get a dotted plot like this. If we get the fitting curve of the decay, we can see the decay constant is around 1 microsecond, which is pretty fast. So we talk about why we want to invention the wide field MIP imaging is because we want to boost the imaging speed. So we tested out whether we can really achieve that. And again, using the same polymer sample as an example, we demonstrate that we can using a single hot and cold frame subtraction to create a contrast like this. Although it's a little bit noisy, but you can still see the weakly focused IR spot in the center. So we can achieve over 1,000 frames per second vibrational imaging, which is a record in the field. We can always increase the averaging speed by, we can always increase the image quality by averaging more frames. So we did that. We also quantify the signal to noise ratio as a function of pixel counts or the photon numbers. And through the fitting, we see that our microscope is operating at near shot noise limited regime. Next, we want to demonstrate that even with the wide field geometry, we can still achieve much improved resolution. So we use two samples. The first is the 3-letter etched off from the thin polymer deposit sample on silicon. So the MIP 3 letters are bare silicon. We did our imaging, and we then quantify our spatial resolution through the sharp edge. We can see that even with the wide field geometry, we can still get 500 nanometer spatial resolution. We also demonstrate that we can do the particle imaging. So here is a 1 micron diameter polymer particle sample. We can get, when we tune to the absorption peak of this polymer, we can get a clear contrast out of this sample. If we tune away from the absorption peak, the contrast is gone, which further demonstrates that we have high spectral fidelity. And we further confirms that using the spectral measurement, we also got very good consistent results with the gold standard spectroscopy measurement. Beyond the polymer sample, we apply it to study the cells. We're using the ovarian cancer cell line as an example. We focus on the lipid peak and the protein peak in the cells, and this is the off-redness peak. So we image at these cells at the imaging speed of 2 Hz. If you recall the previous scanning version, taking one image like this will take about 4 minutes. So our wide field geometry is indeed much improved, provide much improved imaging speed. Yeah, you may ask that if you do live cell imaging, will the thermal heating process cause some damage to the cells? So through theoretical calculation, we calculated that for the live cells, the temperature increase for this type of laser is typically on a few Kelvin or few degree temperature increase, which is considered to be safe and cause minimal thermal perturbation to the cells. So the MIP can be implemented in many other modalities. We can couple a Raman spectrometer here in our MIP setup, and by doing that, we can get a IR spectra and the Raman spectra from the same spot in the same time. The reason why I want to do this is because these two vibrational spectroscopy technique are complementary to each other. So it would be nice that if we can get both information. We can also couple a dark field imaging modality to this so that we can significantly increase the signal to background ratio. We further couple a quantitative phase imaging modality in our MIP setup so that on top of a quantitative phase imaging contrast, we also add additional domain information of the infrared absorption. So as I mentioned before, the IR photothermal is a dynamic process which has heating and cooling. So the previous version, we need to scan the delay between the pump and probe to map the dynamic process. So recently, we've also proved that we can do it with a single shot. Basically, we're using a very high-speed digitizer to digitize the process so that with a single shot, we can map the whole process without scanning. So with all this said, I have introduced the technological aspect of our technology. Now let's move on to see what we can use this technology to study the biological system. Today, I'm going to focus on one important application, that is cell metabolism. The reason why I want to study cell metabolism is because it's very important for cellular functions. It's related to cellular energy reproduction and also cell respiration. And it's also related to many diseases, taking the diabetes, for example, which means that our body has difficulty in handling the sugar molecule, which is the metabolism. Another example is that for Alzheimer's diseases, there's numerous evidence suggests that the metabolic alteration is associated with a disease onset. So despite its importance, it's very challenging to visualize the cell metabolism at the single cell level. There are many existing technologies to do that. For example, the MRI and the PET, we can do a whole organ level metabolic imaging, but the spatial resolution, we cannot see the cellular or even sub-cellular metabolic processes. There's another technology called imaging mass spectrometry, where we can get detailed metabolic products using this technology, but it's destructive and also it takes very long time to achieve single cell resolution. On the other hand, the optical imaging offers a very nice bridge between these two worlds. It offers the sub-micron spatial resolution, but also offers a relatively high imaging speed. And because of many optical imaging modalities, we can integrate them together to provide complementary information. So another important aspect when we talk about metabolic imaging is the high specificity. When we look at the cell image taken with a phase contrast image, we cannot really differentiate the metabolic products with other endogenous molecules. The fluorescence imaging is very powerful in specificity, but we have talked about that the bulky fluorescent labeling will cause the perturbation to the metabolic process. Therefore, we need a label that has a similar performance in terms of specificity, but the label size needs to be much smaller compared to a fluorescent labeling. And the MIP spectroscopy imaging provides a very promising way to do this. So the fluorescence imaging in terms of detection, achieve specificity is because the target molecule, the dyes will emit fluorescence in the new wavelengths. So we know that when we detect at that wavelength, we know that this comes from the product that we targeted in. For the MIP spectroscopy, we also introduced a labeling. Here is the triple N bond, we call AZ bond. Here's this one molecule that is very important to life, that is fatty acids. You can see the labeling is much smaller compared to the fluorescence labeling. What cool thing about this is that the peak of this bond actually lies in a window, what we call sub-cellular window. It's basically like you're generating a new wavelength for us to specifically target this molecule. So here's a typical cell spectrum if we don't have the labeling, and this is the spectrum if we have this labeling. So we can see we have generated new wavelengths. And let's take a look how we do that. Firstly, we have our small molecules labeled with air tags and fatty acids with azides, and then we feed it into our biological systems. It's either a cell or a tissue. Then the cell will uptake this labeled molecules and use that to synthesize macromolecules such as the lipid droplets, phospholipid, or cholesterol esters. So that only these newly synthesized molecules will carry these probes, and we can probe that with our MIP imaging with high fidelity by targeting that new generating wavelengths. And we put it under our microscope, and we can do the imaging. So with this concept, I will introduce you two important applications for metabolic characterization. The first one is the microbiology. The second one is in neuroscience. For the microbiology, I'm going to introduce the rapid determination of the drug efficacy, or more specifically, to determine whether the antibiotics is working or not. The motivation for this project is that the antibiotics resistance is causing a lot of people to die and causing a lot of economic burden around the world. And the major driver for the antibiotics resistance is because the misuse and overuse of broad spectrum antibiotics. And the key way to combat that crisis is to precisely prescribe the antibiotics that will be effective so that we don't have to use a broad spectrum widely. The testing is called antibiotic susceptibility testing, or AST for short. The conventional way or the current workflow to do AST is through culture-based. Well, here's our bacteria sample. We add in antibiotics and we cultured it for a few days. We allow them to replicate. So if the antibiotic is not working, which means that it does not kill the bacteria, the sample will keep grow and form into colonies. And if the antibiotic is working, we will see the colony does not form. It works, but it's not that efficient, and the time actually matters a lot, especially for those patients in acute conditions such as sepsis. But our MIP can do metabolic imaging, so we are wondering, can we monitor the bacterial metabolism instead of monitoring the growth to determine the drug effectiveness? Again, we have our bacterial sample, we have antibiotics, we add them together, and then we don't need culture for a long time, but rather we can directly monitor whether the bacteria is still metabolism or still eat or not upon the antibiotics treatment. The take-home message will be marrying the metabolism is hopefully is faster than marrying the growth in determining the AST. And we achieve this by monitoring the protein synthesis inside the bacterial cells, and the probe we're using is called carbon-13 glucose. So basically, we're monitoring whether the bacteria will uptake glucose and convert that into protein. And the carbon-13 glucose, all the 6 carbon atoms in normal glucose are replaced with a carbon-13 atom, and we're monitoring the protein synthesis at the protein MI1 band, which is around 1660, remember. So if the antibiotics is working, the cells will stop metabolism and will not incorporate this heavier isotope into their cell body. So they're this will be their normal protein spectrum look like. But if the drug is not working, the cell will incorporate these heavier items into their cell body. And because of the heavier items associated with the carbon-13, the protein stretching motion, which is a CO stretching motion, will be slower. So that will generate a new peak corresponding as here. So we can, based on the 2 wave number imaging, and determine whether cells are incorporate the carbon-13 in or not to determine whether the drug is effective or not. Because the bacterial samples are so small, for taking the E. coli as an example, is typically the width is about 0.5 micrometer and the length width is about 1 to 2 micrometer. We have implemented several hardware improvements in order to see that image. We have, for example, coupled a nanosecond laser and a speckle reduction module associated with a nanosecond laser to help us to get a beautiful image. These are from reflection image, and these are from the MIP image, which is corresponding to the protein peak. So with this concept, we have tested the effectiveness of 2 antibiotics. The first one is gentamicin, the other one is erythromycin. And we do the 2 wave number metabolic MIP imaging at the original protein peak and also shifted protein peak. For the gentamicin, we can see that the shifted peak has produced much dimmer contrast compared to the original peak, which means that for this antibiotic treatment, there's no carbon-13 incorporated. So we conclude that the gentamicin is effective for E. coli. On the other hand, for the erythromycin, the contrast from the shifted band is about the same as the original peak, which means that the erythromycin is not effective for this bacteria. And this is within our expectation because we know the action mechanisms of these 2 antibiotics. But we can produce these AIC reports with much reduced time compared to the current workflow. Now let's move on to the second project, that is imaging lipid metabolism in neuroscience. The reason why we want to image the lipids is because in our brain we have a lot of lipids, and it's closely the dysfunction or dysregulation of lipid metabolism is related to many neurological diseases such as Alzheimer's and Parkinson's. So to start with, as a lipid imaging modality, we want to demonstrate that we can indeed visualize the lipid in the cells with very high resolution and high specificity. Again, we're using the fatty acids as a probe. We have our AZ labeling here. This cell is the neuron glioma H4 cells. We image at the total lipids peak, and we image at the newly synthesized lipids peak. So we also did a Bota-P fluorescence imaging. The Bota-P is a dye that attached to the lipids, so it's a very standard lipid imaging modality. If you compare these 3 images, we can see they overlap pretty good. And we also quantify our imaging resolution by pinpointing to a tiny lipid droplets here, and it's about 500 nanometer. If we put side-by-side comparison with conventional mid-IR imaging, here's also a single cell. So you can see with that resolution associated with conventional IR imaging, we cannot see inside the cells, it's just a blob. But using our much improved resolution MIP microscope, we can see very clearly what's going on inside the cells. We can also do spectroscopy measurement on these lipid droplets. So with this concept demonstrated, we then move on to investigate whether we can apply this system to study the lipid metabolic alteration associated with different diseases. And we focus on one gene, it's called GRN, and the loss of function or knockdown of this gene is related to frontotemporal dementia. But there's no imaging evidence to show that how does the lipid metabolism change for this gene loss of function. So we test our sample, which is iPSC induced pluripotent stem cells. We get our control cell length, we have our knockdown cell length, and we image at different wave numbers and also get a ratio image from these 2 lipid imaging. We also get a histogram from the ratio image, and we can clearly see there are 2 populations, one associated with control cell line and the other associated with the GRN knockdown cell line. And for the GRN knockdown cell line, we got a higher mean value, which means that for the GRN knockdown, it has higher lipid metabolism, which is consistent with other biological approaches such as transcriptomic studies. But here we are adding additional imaging evidence to support that. We then move on to study a more sophisticated sample, which is a brain organoid. The brain organoid is a 3D tissue culture where different cell type cells assembly into a blob, and it can mimic the human brain development. It's a very important tool to study the human brain development as well as for certain diseases. So we're adding our probe throwing our probe into the brain organoid cell culture, and then after 24 hours of incubation, we fix our sample and thin slice it, and then do the immunohistochemistry to for two cell types. One is the TUJ one for the neuronal marker, and the GFAP for the astrocyte marker. We then focus on one smaller region of interest and perform the MIP imaging here for the newly synthesized lipids. So from this image, we can see that the contrast is not uniform across the field of view. If we overlap with our fluorescence imaging, we can see it's highly overlapped with the red contrast, which is the astrocyte cells. This imaging evidence is also supported by our spectroscopy measurement, where the astrocytes have a higher AZ signal corresponding to higher lipid metabolism. And this actually supports many other biological discoveries that the astrocytes in our brain plays a very important function in maintaining the cell function.
Can you please explain what's a brain organoid?
Yeah, it's for conventional planetary cell culture is 2D, but for the brain organoids, it's like a 3D culture. So you have a blob of cells that is a few millimeter in diameter, and in the cells in this blob, you have many different cell type, such as neuron, astrocytes, which similar to our human brain. So that when you sort of get everything together, they will actually start to mimic the human brain development, and you can see the layers of different cells that is very much similar to a baby, to an embryo. Yeah. So yeah.
And these things come from a human brain or an animal brain?
So
or something else?
Yeah, actually that's a very good question. As you can see here, is called we call it iPSC. It's actually from you can get it from mouse or from human. So from human, you will call human-induced pluripotent cells. You get a skin cells. You can reprogram it to be like a stem cell, like in the embryo. Then you can use that reprogram the cells to get the 3D brain organoid culture. So people are using it for personalized medicine because you can get a skin cells and then you can grow up like a tiny brain in dish. Then you add in drugs and then to see whether the drug works for the patient or not. Yeah.
One other question. What's the lipid metabolism? That's as opposed to glucose metabolism? So they consume lipid for food?
Yeah, that's also a very good question. So here we are feeding fatty acids, which is a small molecule. And the end products we are detecting actually is lipids. It's like a bunch of fatty acids assembling together to form like a cell membrane, if you remember the double layer structure, or lipid droplets, which is energy. It's a little dots that in our cells to store, basically fat. Yeah, so we are detecting the end products, but we can also do other metabolism. For glucose metabolism, people are using glucose to synthesize all sort of stuff. So it's called like de novo synthesis. We can use glucose to synthesize lipids. We can use glucose to synthesize protein. But here we are using, it's a direct uptake compared to de novo synthesis. Uptake fatty acids synthesis lipids. On the contrary to uptake glucose synthesis lipids. So to summarize my talk, I have covered the technical part of this technology and introduced MIP as a sub-cellular imaging method with high specificity and high resolution. I've also covered two important applications for metabolic characterization, but our technology can do much more. We can apply it to different biological systems for different purposes. For example, we can use it to study brain disease such as Alzheimer's disease to see whether the brain is consuming more, say, glucose or reducing glucose associated with the disease. And we can also do tissue imaging for such as disease diagnosis for histology purposes. So most of the work I present today are done within my previous lab at Boston University or UCSB, and I also like to thank the NIH for support and thank our college and also University of Arizona for the sponsor. Yeah, with this said, I'd like to take any questions that you may have. Thank you. Go ahead.
You mentioned that you are attaching these very small molecules to the bacterial cells or whatever
Yeah,
to do photothermal imaging,
Yeah.
right? But single one of those small molecules is not enough to give you a signal.
yes.
So how many of them do you need to get a reasonable signal?
That's actually a very good question because that's related to your detection limit of your technology, right? So for this setup, we are far from single molecule imaging. So the current limit for this technology is on the order of few hundreds of micromolar. Yeah, but you can convert that into how many molecules based on the molecular weight. Yeah.
So it's still smaller than something like green fluorescent.
Yeah, for fluorescence imaging, people can do single molecule, right? You with a single that protein you can visualize in cells, but for us we don't have that sensitivity. But that's definitely one of the direction that we want to pursue, to further push down the detection limit so that we can see much dilute signal in the cells.
So do you see or have you considered, are there any pathways for translating this into like an endoscopic instrument where it's being used in vivo?
Yeah, that's definitely I've been thinking about it, especially with all the experts in the college who is doing, endoscope or like pen type of stuff. So I think this technology will be very good because it will be impossible for a conventional mid-IR imaging because you're shining light in, there's no light bouncing back. But for us, we have a chance if you shine two beams together, the gray will scatter back so that you can detect. So I think that's opens a very good chance for us to do it in endoscope studies. Yes.
Little brain organoid. Is that what's called artificial intelligence?
I cannot say that, but I mean, people are actually reading from my previous book at UCSB. They are culturing these organoids on electrodes, and they are reading the neuronal firing patterns from this organoid. So, which means that the neurons are response to external stimuli that they are communicating with each other. But does that mean anything? I guess researchers are still studying that.
You mentioned about increased data resolutions. I think that how about actual resolution because the resolution.
Yeah, that's also a very good question. So from the previous 3D reconstruction image, that one. So yes, the Z resolution is also increased compared to the mid-IR imaging because the Z depth is also related to your wavelengths, right? So we can now focus tighter using the visible light, so we can achieve tighter focus in Z compared to conventional IR imaging. We also quantify that using this setup, so the X-ray resolution will be few hundreds of nanometer, and the Z resolution will be few micron. Yeah, just as what you will expect from the visible microscopes.
What is the penetration depth of your imaging technology? Can you also comment about the impacts from the photon scattering?
Yeah. So two questions. First one is the penetration depth. I think you have two laser beams, right? So it's depending on both of the beam. I think it's probably suffer more from the infrared light because depending on what sample you're studying, say we will study biology, majority of the sample is water, and the water is a very strong absorber for infrared region. So if you want to focus your IR light in the water absorbing peak, is probably less than 10 micron penetration depth. And I guess that's echo back to the endoscope thing because the penetration thing. But if you tune away from, say, this lipid peak, as you can see, from 80, as deep as 80 micron, we can still get some contrast. It's all depending on which absorbing peak you are using for the infrared. Yeah, for the visible light, the scattering will definitely be an issue for us, but The good thing I think is that because we are detecting the difference between two scattering states. So, hopefully the background scattering is not changing too much, but rather the error absorption induced scattering, we can still get it out. But so far, most of our sample we only focus on the either 2D plenary culture, cell thickness is only few micron, or a very thin tissue size is also few micron. So we don't encounter that problem too much. But if we want to push it to, say, unprocessed thick tissue, the scattering thing is definitely an issue. Yeah.
What's the physical mechanism of your contrast in the visible? Is it the change of refractive index?
I think it's both because the thermal will cause expansion, and the expansion, the dimension when your volume is bigger, then the index will be reduced. So we sometimes we call this technology thermal lensing effect. Basically, it's your index is changing. Yeah.
So that's the only thing that you consider the change of refractive index due to the change of volume.
I think that's a major contribution, but there are also different mechanisms and also for different samples different. Say you have a thin film sample, I guess the index change is straightforward to understand because the interface reflection is changing. But if you have a particle sample, then your scattering is changing, and it's also related to what medium you are embedded in. So the index mismatch and also the thermal gradient is also changing. Yeah, so it's for the exact mechanism, we are still trying to model that. Yeah, but that's also a very good question.
But for a few degrees Kelvin change of temperature, the change in refractive index cannot be that great.
Actually, the relation is 10 to -4. So, but if you increase 10 degree, then it will be 10 to -3. We can detect that. Yeah, if you think you detecting Raman scattering, the scattering cross section is 10 to -8, people can still extract that. But for us, we are detecting 10 to -3, so we can detect that. Yeah.
Any more questions? Last question. If not, let's give applause to the.
Yeah.